Okay, so again, chapter 11, muscular system. So there are over 600 muscles in the human body. That is a whole lot of muscles. In this lab, we're just gonna be looking at about 10 to 15 muscles. So we're not even getting close to the amount of the muscles in the human body, but there are some prime muscle groups that are generalized and viewed as most important. When we look at muscle contraction, we see that there is always an agonist and an antagonist. The agonist is going to be the muscle that causes the movement. You might also hear it called the prime mover. And the antagonist is a muscle that acts in an opposite way to the agonist. So for example, if I was flexing my biceps brachii, which is, the biceps, my biceps would be the agonist and my triceps would be the antagonist, because when my bicep contracts, my tricep relaxes. So there's always going to be an agonist or a prime mover and an antagonist or a relaxer. We also have synergists, which assist the action of an agonist. So like with my biceps brachii, the deltoid might help the shoulder muscle, the deltoid might help a little bit. You have more muscles in your lower arm and in the forearm also, that would help with that movement. And then lastly, we have origin and insertions. Origins are where the muscle attaches. An insertion is where it inserts. So it originates and then it inserts. And luckily, we will not have to learn the origins and insertions this semester. In past semesters, I've had my students learn origins and insertions, which gets really complicated, but we're not going to do that this semester, so relax. So this is nothing I'll quiz you on. This is just a reference slide, this next two. But just for reference, whenever you hear the name of muscles, a lot of them are named based off their location, their size, their shape, the direction of fibers, their origin, and their action. So the ones that are named off their location are pretty obvious. Anything in the abdomen would be the abdominis, like we'll look in a little bit. Our abs are called the rectus abdominis, so the abdominal muscles. Brachium is arm, so we have the biceps brachii. We also have the biceps femoris. So femoris is thigh. So it's great to know that brachium is arm, femoris is thigh. You can think femoris femur. Gluteus is going to be the butt, and pectoralis is going to be the chest. We can also name muscles based off their size. Brevis would be short, longest, long, maximus, largest, minimus, smallest. And then shape, we won't be looking at much that deal with shape, but those are the three you might hear named based off their shape. We also look at the direction of the muscle fibers, so whether they're straight or parallel, whether they're transverse at right angles or oblique slanted. That's not as important. One that really is important is their origin. So anytime you hear bi, tri, or quad, that means 2, 3, or 4. So like our biceps have 2 muscles or 2 origins. Our triceps have 3, and our quadriceps have 4. So anytime you hear bi, associate that with 2 muscles. Tri, 3 muscles, quad, 4 muscles. And then lastly, based off their action. So an extensor would be to open an angle, a flexor would be to close an angle, an abductor would be to move away from midline, abduct from midline, and adductor would be to add towards midline. Again, those are just for reference if we're ever looking at a muscle name and you're like, ooh, pectoralis major, what's pectoralis again? You can go look at that and see, oh, pectoralis is the chest. So now we're gonna get into muscle locations. So this is when it's gonna be important to kind of maybe feel them on your own body, maybe do some flexing right now, and make sure you can locate where these muscles actually are. Another thing I like to do during this lab is, you know, maybe throw some workouts in the chat as we go through. So whenever we get into the body muscles, we're gonna start with the face and neck. So probably you won't know many workouts when it comes to those, but like when we look at the biceps brachii, if anyone knows like a good exercise for the biceps brachii, throw it in the chat just so in case anyone isn't familiar with it, they can think, oh yeah, well if I, you know, do a bicep curl, that's what it's working at. So feel free to throw some exercises in the chat. So here is kind of a broad overview of the muscular system. You can see how many muscles are in the body, and this isn't even all of them. There are hundreds and hundreds of more muscles, and that blows my mind that there are 600 in the body. But luckily we were just learning a few. So here's kind of a broad view, but we're gonna get into the more detailed pictures. So the first muscle we're gonna look at is one of the facial muscles, the temporalis. This muscle aids in chewing. So whenever you chew, you can feel this muscle contract. So if you sit here right now and simulate chewing, you're gonna feel a muscle contracting about right here. That is the temporalis. It aids in chewing. It sits right next to your temporal bone, right on your temples, so that makes sense. Temporal bone, the temporalis, that's the muscle that aids in chewing. Then one of my favorite muscles is the sternocleidomastoid. It's a big word, but it makes sense once you break it down. So if anyone remembers this bone in our chest right here, this is the sternum. It forms our breastplate or our chest plate, sternum. And then if you also remember when we looked at the bones of the skull, when we were looking at the temporal bone, we had that process right here called the mastoid process. So the sternocleidomastoid is a muscle that attaches or inserts at the sternum and attaches at the mastoid process. And this is the muscle, it's on two sides, that helps us nod our head. So the sternocleidomastoid goes from here to here, forming an upward V. And that is the sternocleidomastoid. Sternocleidomastoid. Sternum to the mastoid process. And again, it helps us with head nodding. So for example, whenever you take your practical, that would be one you probably want to study a few times. Practice how to spell it, how to write it. On your practical, I won't, count off if you put an I before an E or put an A instead of an E, but I do need it to sound like how the word is pronounced. So if you put sternocleidomastum, I would maybe give you half credit on that, maybe not. Let me see, pull my chat up. And then whenever, if you put sternocleidomastoid and spelled the cleido wrong and the mastoid wrong, that's fine. As long as that prefix and suffix, I can tell you can say the word, that's good. But, practice writing the longer ones a couple times. So yeah, let me get this pulled up. So, yeah. We'll go over trapezius when we look at the back. So this is the anterior view of the front of the body. So you can see that sternocleidomastoid attaching right there at the manubrium of the sternum, right there at the top. And so now we'll get into the arm. So the muscle of the shoulder, this is the deltoid. Anyone know any good deltoid exercises? And then Olivia asked, we only need to know the locations, right? Or do we need to know, for example, if it causes head nodding or anything like that? Location is key. For the muscles, I have you identify pictures of them. So, the function like head nodding, chewing, things like that are kind of just to supplement your understanding of them. But I don't think on the practical I ask like what muscle aids in head nodding or what muscle aids in chewing. So with the muscles, location is key for the exam. Good question. So yeah, Marina said lateral raises. So yes, lateral raises are great. You know, IYTs are what I do for deltoids. So I hold 2 free weights in my arm. I go I, Y, T. Any type of like upward movement will work with your deltoids. So those are great exercises there. And again, your deltoid is the small muscle that sits on top of your shoulder. Then we have the pectoralis major, so your pecs, and on the practical, you need to put the muscle name. I won't take like pecs or delts or abs. I need to know the muscular name. So a lot of people go to the gym and they're like, yeah, I'm gonna work out my pecs, but whenever you're like, what muscle is that? They're like, the pecs. No, it's the pectoralis major. So make sure you know these full names. So the chest muscles are the pecs. Anyone know some good exercises for pecs? Pecs exercises? Yes, bench press is the most common exercise. So I bench pressed today and oh, I am so sore in this entire area right here. So bench press, pretty much any type of pushing is gonna work your pecs. So if you, you know, do push-ups, anything pushing is gonna be a great pec exercise. Then we have the biceps brachii. So we all know the biceps, but you need to know that the biceps in the arm are called the biceps brachii. One of the muscles of our quadriceps is called the biceps femoris. So if you just say biceps on the exam, I could think, oh wait, are they talking about the biceps of the leg or the biceps of the arm? So make sure when you're talking about the biceps and the triceps, you say biceps brachii, triceps brachii, because you also have biceps and triceps in your leg. So biceps brachii, this one in your arm. Anyone have some exercises in here? I've got to have some fitness people in lab or ESS majors. Anyone that works out? Anyone know some good biceps? 21s, I do not know what a 21 is, and I do not know what preacher curls are. So these are good because I'm actually doing I'm in a fitness goal mindset right now. I work out like 4 times a week, so I'm always looking for new exercises. So 21s and preacher curls, I will look into those. Interesting. And then, you know, any type of like curl movement here. So any type of curl for the biceps is great. So we'll continue. So now we are on to the posterior side. So again, we have those delts, our upper back muscle, our traps. This is the trapezius. So our traps are the trapezius. So anyone know some good like trapezius exercises, some good traps exercises? Traps. Here. Shoulder shrugs. Shoulder shrugs are great. So, you know, holding some free weights in your arms and, you know, shrugging like that. That is a great traps exercise. So that's going to work those muscles of your lower back or of your upper back, these right here. Cool. Then we have the latissimus dorsi, so the lats. That's what you hear them called most commonly, the lats, but they are called the latissimus dorsi. If you remember, dorsal means the posterior side, so the latissimus dorsi, the back muscles. And these are the mid and lower back, so these are your lats. Any good exercises here? For latissimus dorsi, anyone know any for the lats? Grip pull down, wide grip pull downs, yes. Pull ups, yes. Lateral pull downs. So any type of pull is going to be great for the lats. So you know, rows are great for the lats. You know, there's that machine at the gym where like you start up and you pull it down, and you pull it down. Any type of pull is going to be good for the back of the body. Any type of push is going to be good for the front of the body. So pulls are good for back, push is good for front. And that's not anything I'll test you on. It's just good to know that for whenever you get into your more major related classes like XPhys, resistance training and conditioning, things like that. Or if you're just working out on your own and want to know. Just a fun fact, you can improve your posture immensely by doing more pulling. So a lot of people go to the gym and they do primarily push exercises. Like they get to the gym, they bench press, they squat, they do that, and they kind of forget pull. So they kind of leave out the posterior muscles, they leave out the hamstrings, and it leads to imbalance in your muscle mass. So it's always great to do pulls, and whenever you do a lot of pulls, your posture will improve. If you do a lot of pushes, you're going to constantly, your muscles are going to be a lot bigger in the front, you're going to start shrugging forward. So it's great to balance your push with your pull. You don't want to do more push, you don't want to do more pull, you want to have even amounts so you don't get imbalances. If you imbalance if let's say you only squat for legs and you never do any hamstring, your quads will dominate your hamstrings, and that can lead to a weakness in the anterior tibialis, which can lead to a tibial tear, ACL tear, all of that. So you always want to balance your exercises. We're going to go ahead and skip infraspinatus, which is just one of the muscles of the shoulder blade, but we're going to go ahead and skip that. I was going to say we had more. When looking at the core, we have two muscles that we're going to focus on here. So first we have the rectus abdominis. These are our abs, the rectus abdominis. And a way that I remember that is I know for me, anytime I do abs, I feel rect afterwards. So rectus abdominis, rect. You rect your abs, rectus abdominis. And again, that's one that people are like, yeah, the abs, I know where the abs are. But then when you ask them like on the exam, like, identify this muscle, and they're like, abs, but crap, what's the like muscular name? Rectus abdominis. And then we have the external obliques. So the external obliques are these muscles of your side. I forgot to ask for abs exercises. Abs, you can do, crunches, Russian twists, any type of planks, things like that. External obliques, if you put a dumbbell in one hand and then kind of, did side to side, movements, things like that. You could get a band and do pulls, cross body pulls. Those are great for external obliques. External obliques. Lateral pull downs are also great for obliques, and I never really put that together until I just recently started using the lateral pull down machine in my apartment's gym, and oh my gosh, I feel that way more in my obliques than in my pecs, which is crazy. So yeah, lateral pull down if you want to improve your obliques. Okay, and so this is a diagram that actually I took from the respiratory chapter of our book. But respiration is one of the coolest functions of the body, in my opinion. So we're going to look at the two muscles that are involved in breathing or respiration. We're not going to look at the different functions that are happening here. But we have two major muscles involved with respiration or breathing, and that is the diaphragm, which sits right here. It separates the thoracic cavity from the abdominal cavity. It sits right below your ribs. And then we have the external intercostals, which are the actual muscles of the ribs. And what happens is whenever you breathe, our lungs sit right here. Our lungs are actually giant. They're like that big. And they sit right here. And when we take a breath in, our diaphragm pulls down. So you go. So that's what the diaphragm does. Whenever you take a breath in, it goes down and it expands the lungs. With the intercostals, which anytime you hear the word costal, that means ribs. So like the costal bones or the rib bones. The external intercostals are these muscles that sit on the outside of the ribs. And whenever you breathe in, they pull out. So they go. So together, the diaphragm and the external intercostals move like this. Sorry, I'm such a breathing nerd, but we'll save, you know, the volume and the pressure, the movement down, the movement out for respiration, but those are two muscles. A lot of people think they're like organs or something, but the diaphragm is a muscle and the external intercostals are a muscle. So know those. So I think this is the last slide of the arm that we look at. And all we're going to focus on here is the triceps brachii. So again, we have the biceps brachii in the front, and the back we have the triceps brachii. So it's very hard to see mine because I barely have one, but I'm working on it. And there we go. You can see it like kind of right there. I have been on a very strict workout routine for the last 5 weeks now. And when you take exercise physiology, you learn that in the first 4 to 6 weeks of resistance training, you only will see neurological changes. So you'll only see changes in how your brain communicates with your muscles. So you'll see improvements in form, you'll see maybe small increases in weights. But after that 6th week is when you actually see changes in your muscle mass. And that's a big issue on why people give up on exercising after a couple weeks, because they're like, I'm not seeing any results. Well, it takes 4 to 6 weeks for you actually to see changes in your muscle mass. Those first 4 to 6 weeks are all neurological changes. So your brain making connections with your muscles, things like that. So don't give up, because I'm like 4.5, 5 weeks in and I'm like, why am I not getting stronger? But it's coming. So yeah, we already went over biceps, and again, you want to make sure you label biceps brachii and triceps brachii, because we have biceps femoris and triceps femoris. We're not going to look at subscapularis. Again, that's one more muscle of the shoulder blade, or scapula, but we're not going to look at those. So now we're getting into the leg, the lower body. So we have the glutes, we have the gluteus maximus, which is the big muscle, and the gluteus medius, which is the upper smaller muscle. So the gluteus maximus, what can you do exercise-wise to improve your glutes? Glute exercises. This is a super easy one. Like one of the most basic squats. There we go. So squats. Squats are the main glute exercises. Band exercises are great. I had a student in my lab on Tuesday show how she puts like a band on her like thighs right here and then does like side steps for her glutes. So glute exercises, very important. Fire hydrants, never heard of that. I'll have to look into that. Fire hydrants. Very interesting, because I'm always looking for glute exercises. So fire hydrants, I'll look into that. Cool. So now we're gonna get into the muscles of the actual legs. So those were kind of the butt area. Now we're into the legs. So note this down. This is important. It is not written anywhere on your slide. The sartorius, you can see it right here, is the longest muscle in the body. The sartorius, the longest muscle in the body. It inserts right here. You don't have to write this down. This is just for reference. It inserts right here at the anterior superior iliac spine, the ASIS, which is that point right there. And then it attaches down here at the medial tibia, so all the way down at the lower leg. So this muscle moves all the way from your external hip. Move some things around. External hip all the way into your internal knee. So it's a super long muscle. There we go. So sartorius, longest muscle in the body, goes from your hip to your internal knee. Next to the sartorius, we have the gracilis, which is a big muscle of the inner thigh that is located right here. If you go to the gym, you can find a lot of machines that focus on the gracilis, like the hip abduction and adduction. Those will work your gracilis pretty hard. And Then we have the quadriceps. So again, there are 4 muscles of the quadriceps. Select turn motion activated. So we have 4 muscles of the quadriceps. We have the rectus femoris, the vastus medialis, the vastus lateralis, and another one that's not listed on here. But I'm not going to have you know the 4 muscles of the quadriceps. I just want you to know that there are 4 muscles in the quadriceps. You don't need to know the rectus femoris either. Here you can see the hamstrings. We have 3 muscles of the hamstrings, which I think I have listed on the next slide. But again, you can see that biceps femoris. So like we have the biceps brachii in the arm. We have the biceps femoris in the leg. And then of the shin area, we have the tibialis anterior. So if you remember, the tibia is this bone right here. The tibialis anterior is the bone or the muscle that is on the front side of the tibia. If any of y'all played soccer or were dancers or anything like that and got shin splints when you were younger or recently, that would be cramps of the tibialis anterior. So shin splints deal with the tibialis anterior. Most of the time I've always heard it called the anterior tibialis. So if you hear me say anterior tibialis, same thing. And then lastly, we have the gastrocnemius, the calf muscle. So this is the gastrocnemius. And that's one of those ones where on the exam, you're like, oh, I know that's the calf, but what's the name of it? Gastrocnemius. Why do we call it the calf? I have no idea. It is such a random name because you would think like calf, like baby cow, but who knows, because it doesn't sound like gastrocnemius either. Super random. But yes, the calf muscle is our gastrocnemius. And that's so cool, like if you're ever working out with your friends or anything or walking up the hills on campus, you'd be like, oh, my gastrocnemius is so sore. And they're going to be like, what? And you're going to be like, I'm an exercise sports science major. I know my muscles. And it's just cool. So. All right, so lastly, we're gonna look at the muscles of the hamstrings. So we have 3 muscles of the hamstrings, and I want y'all to memorize these. We have the biceps femoris, the semitendinosus, and the semimembranosus. And a way I remember these is the saying, butchers tenderize meat. Butchers, biceps femoris, tenderize, semitendinosus, meat, semimembranosus. Meat, mem, tenderized tin, biceps femoris, butchers. So biceps femoris, butchers, semitendinosus, tenderized, semimembranosus, meat. Those are the 3 muscles of the hamstrings. I won't have y'all identify a picture of them, but I might ask like, a question saying, give me the last of the, whatever. So I might list 2 of them and say, which one am I missing or something like that. So know the muscles of the hamstrings. So those are all the muscles we're gonna learn. I'm gonna go through them really quickly. You can or cannot or whatever, use the chat to answer these. I'll go through the muscles of the body. I think recalling them right now is gonna be really helpful in your memorization. So again, you can unmute yourself and answer, or you can just answer to yourself, whatever you want to do. So first, we have this muscle on the side of the head attached to the temporal bone that aids in chewing. Does anyone remember what this one is called? This muscle that aids in chewing. This is the temporalis. Yes, the temporalis. So this is the muscle that sits right here and aids in chewing. Then we have a muscle that kind of forms a V shape right here and helps us with head nodding. This is the what? Head nodding. Yes, this is the sternocleidomastoid. Sternocleidomastoid process. And this aids in head nodding. Then we have the muscles of the chest. What are the chest muscles called? These right here. Yes, these are the pectoralis major. Make sure you know the pectoralis major. So these are your pectoralis majors, the chest muscles. What about our shoulder? The muscle of our shoulder, right here. Deltoid, yes, the deltoids, the deltoid. This muscle right here, the upper. Biceps brachii, yes. Good job for adding the brachii. A lot of people forget to put brachii and get it wrong. So biceps brachii. Posterior one. Triceps brachii. Yes, your triceps brachii. Muscle of the upper back. So up here. Upper back muscle. Trapezius, yes, your traps, your upper back muscle, trapezius. What is our mid and lower back muscle? Latissimus dorsi, yes. So your lats, your latissimus dorsi are your mid and lower back muscles. What is the big butt muscle? Gluteus maximus, yes. And then the smaller one on top of it is the gluteus minimus. But the gluteus maximus is the one that is key to know. Then we have the longest muscle of the body that goes from your hip to your internal knee. What is that? Longest muscle of the body. Yes, the sartorius, longest muscle of the body from the hip to the knee. That is our sartorius. Next to it, like right here, good hip abductor. Anyone remember what this one's called that sits kind of next to it? It's the gracilis. So the gracilis, yes, the gracilis is right in here. And that's a good exercise if you do like hip abduction. What is this muscle group, this entire muscle group right here called? There's 4 muscles here. What is this 4 muscle group called? It's an easy one. Yes, the quads, the quadriceps. Posterior side, what are these muscles called? There's 3 of them. Hamstrings. Oh wait, no, sorry, hamstrings. And again, we have 3 muscles of the hamstrings. Butchers tenderize meat. Biceps femoris, yes. What's the next one? Tenderize, butchers tenderize. Biceps femoris, semitendinosus, and meat. Semimembranosus, yes. So butchers tenderize meat. Biceps femoris, semitendinosus, semimembranosus. And then lastly, we have the lower leg. What is the muscle of the shin called? Definitely feel it if you do any type of plantar or dorsiflexion. Yes, tibialis anterior or anterior tibialis. And then lastly, we have the calf muscle, which is called the gastrocnemius. Okay, good job, y'all. So, again, a lot of us know, the calf, the abs. Oh, I forgot to do abs. Abs, rectus abdominis, and then, the external obliques. But a lot of people know the abs, the quads, the biceps. Make sure you know the full name, the rectus abdominis, the quadriceps, biceps brachii. So that is muscular. So if y'all have any questions, I'm gonna go ahead and start moving on to our next slide. Feel free to ask me anything you need while I'm headed that way. All right, screen share. All right, cool. So this is chapter 12, I believe, the anatomy and physiology of the neuron. So from what I heard from my Tuesday lab is that y'all have started on the nervous system in lecture. So hopefully this isn't all new to y'all. Hopefully it's kind of familiar. But feel free to stop me if I'm going too fast or if you get lost or anything. So let's begin. So the nervous system. What is the nervous system? The nervous system is the main controlling and communication system of the human body. So it's how our body communicates with itself. It involves three major functions, which are sensory input, sensing something, integrating it, deciding what to do with it, and then motor output, actually causing an action. So for example, a bee lands on my arm. I will sense that. Sensory input will be sent to my spinal cord and my brain. My brain will decide, oh, bees are scary, and it will cause a motor output of my arm pulling away. So sensory input is the bee landing on me and the nerves in my arm sensing that something landed on my arm. Integration is that sense being sent to the spinal cord and then to the brain, and my brain integrating, oh wait, we're scared of bees. And motor output is an impulse sent to my biceps to pull my arm away. So motor output's the action, sensory input is the sensing, and integration is just deciding what to do with it. So when we look at the nervous system, we have two major divisions, but these divide much further. So we're going to make a graph. So let's begin. So I think this is a fill-in-the-blank graph on your slide, and I'm going to add some more information to it. So I'm going to stop sharing my screen and use the whiteboard. So if you want to get a piece of paper out, or however you take notes, I'm going to do a little diagram here. I forgot all my pretty colors upstairs, so I'm just going to be using the colors that are down here. So we have the nervous system. So what are the parts of the nervous system? Well, we have the brain, we have the spinal cord, we have all the nerves that come off of that. So we need to make it into divisions to be able to classify the individual functions and characteristics of that. So to do that, we first divide the central nervous system from the peripheral nervous system. So the central nervous system, the CNS, versus the peripheral nervous system, the PNS. What makes these two different? The central nervous system is your brain and your spinal cord. The central nervous system is your center, your brain and your spinal cord. The peripheral nervous system is all the nerves branching off of the brain and spinal cord. So all the nerves of the periphery, or your brain and spinal cord. So of your periphery, that's talking about the arms, the legs, the hips, stuff like that. So the central nervous system is your brain and spinal cord. The peripheral nervous system is all the nerves branching off of the brain and spinal cord. So your brain and your spinal cord aren't in your fingertips, but you have nerves that connect to the spinal cord. So anytime you have sense, anytime you touch something, the nerve in your fingertip will sense that and send an impulse to your spinal cord, send it to your brain, integrate it, and then, let yourself know, oh, I'm touching the whiteboard. Oh, I'm touching the desk. Oh, I'm touching my hand, things like that. So nerves are part of the nervous system. So we're going to divide this even further. We're going to divide the peripheral nervous system. So we're going to divide the peripheral nervous system even further. The central nervous system, that's the brain and spinal cord. Now we're going to get into the peripheral nervous system. So we're going to divide the peripheral nervous system into the motor division and the sensory division. So the motor division and the sensory division are our two divisions of the peripheral nervous system. Our motor division is called the efferent division. So motor, efferent. Sensory is afferent. Whenever you hear the terms efferent and afferent, afferent is arrive. It is arriving at the brain and the spinal cord. Efferent is its exiting. So like how we saw at the beginning, we have sensory input, integration, and motor output. Sensory input is an afferent impulse. The impulse that we're receiving is an afferent impulse. It is arriving at the spinal cord and brain. The brain and spinal cord integrate it, and then we have a motor output. An efferent impulse is sent. Efferent exit. It is sent to the muscle, to the organ, whatever part of the body we need an action to occur. So efferent exit, afferent arrive. And I have a saying that we'll look at in just a second that will help you memorize that. So now we're going to divide the motor division even further. So our motor division, let's divide that. So we're going to divide our motor division into the somatic and the autonomic. So autonomic and somatic divisions. Autonomic nervous system is our involuntary control. Involuntary control. So this will control our heart rate, our digestion, the smooth muscles of the digestive system, the cardiac muscle of the heart. Autonomic is involuntary. Autopilot is how I remember that. Autonomic, autopilot, it's doing it on its own. It's involuntary. It does not require any type of control. Somatic, however, is voluntary. Somatic is voluntary. So this is looking at our skeletal muscles, what we have control over. So autonomic, involuntary, you can think autopilot. Somatic is voluntary, skeletal muscles. We have one more division to make, and that is of the autonomic nervous system, our involuntary nervous system. And we divide that into our parasympathetic nervous system, parasympathetic, and our sympathetic nervous system. What are these? Our parasympathetic, or I'll start with sympathetic. Our sympathetic nervous system is our fight or flight response. So sympathetic is our fight or flight. Parasympathetic is our rest and digest. So, for example, all of us right now are most likely utilizing our parasympathetic nervous system. Our bodies are just chilling, everything's normal. However, let's say a pack of wild dogs just ran into the room you're sitting in right now. Your heart rate would go up, your pupils would dilate, you would salivate much less, you would get dry mouth, you would start breathing heavier, you would be scared. That's an impulse due to the sympathetic nervous system, our fight or flight response. A way I remember these two is you can think parasympathetic, rest and digest. Parasympathetic, you can think paralyzed. So if you were like paralyzed, you would be doing a lot of resting and digesting. Sympathetic, you can think like, oh, they were really scared, so I feel sympathy for them. So rest and digest, parasympathetic, paralyzed. When you're paralyzed, you will probably rest and digest a lot. Sympathetic, fight or flight. They were scared, so you have sympathy for them. Silly, I know, but that's how I remember it. So one last time, I'm going to go through these just to instill it in your minds. The nervous system has two major divisions, the central nervous system and the peripheral nervous system. The central nervous system is our brain and spinal cord. The peripheral nervous system is all the nerves branching off of the brain and spinal cord. We divide the peripheral nervous system into the motor and sensory divisions. Our motor division is our efferent impulses, so impulses exiting the brain and spinal cord. Our sensory afferent division is all the impulses arriving at the spinal cord. We divide our motor system, or our motor division, into two further divisions, the autonomic nervous system and the somatic nervous system. The autonomic nervous system is our involuntary control, our autopilot, and the somatic nervous system is our voluntary control, so like our skeletal muscles. We divide our autonomic nervous system into two last systems, the parasympathetic nervous system and the sympathetic nervous system. The parasympathetic system is our rest and digest. You can think paralyzed when you're paralyzed to do a lot of resting and digesting. Sympathetic nervous system is our fight or flight. You feel sympathy because they were scared, fight or flight. So those are the divisions of the nervous system, crucial to know. So I might put this exact, let me make sure I'm screen sharing again. So how you could see this on your exam is I would put this chart and I'd say fill in the missing segment. So, you know, like sensory would be not filled in and I'd have you fill that in. Or I'd ask you, you know, which division of the nervous system is our fight or flight response? Which division of our nervous system is afferent, dealing with afferent signals? So the little notes I wrote below these, brain and spinal cord, nerves of periphery, efferent, afferent, involuntary, voluntary, rest and digest, fight or flight. You want to know those two. Up in case anyone asks me anything. All right, so those are our divisions. So we're going to super briefly look at innervations, not too much, just because we have so much to do in today's labs. We're not going to deeply look at the nerves. We're kind of going to zoom through this. So this is kind of just like a picture of the nerves of the body. You can see the brain up here, you can see the spinal cord right here, and these are all the nerves that kind of branch off and go to different parts of the body. So you can see at the top of the spinal cord, we have a lot of nerves that control our eye, our salivary glands, our heart, lungs, stomach, all the way down to genitals, things like that. And yes, those are the nerves branching off. When we look at the spinal cord in just a little bit, we'll see that it has this end section that will deal with the nerves of the leg. But you definitely won't need to like know this picture or anything. It's just, you know, for fun. So when we look at the nerves of the parasympathetic nervous system, we have cranial nerves and we have spinal nerves. That's all I'm going to go into. We have cranial nerves and spinal nerves. You really don't even need to know that, but yeah. I won't be asking any type of questions about nerves on the practical. So this nerve structure, you're not responsible for this either. You know, we have coverings of the nerves. The epineurum is the outer, perineurum is the middle, endoneurium is the innermost layer, but I'm not going to have y'all understand that. So let's get into the cells of the nervous system. So we have two major types of cells in the nervous system. We have glial cells and we have neurons. Neurons are truly the main cells of the nervous system because they're the ones that actually have that communicative role of transporting impulses in our body. So they're the ones that, you know, you touch a hot stove, a neuron's gonna sense that and send it to your brain and have you pull your hand away really quick. Actually, that will be a reflex, which we'll look at in a little bit, but you touch something kind of cold,
a neuron that extends all the way down to your fingertip. We'll send that all the way to your spinal cord and then to your brain. So neurons are the main, and we'll look at their structure in just a second. But we also have glial cells, and their main function in the central nervous system is to produce cerebrospinal fluid, CSF. When we get into the brain and the spinal cord, specifically, we'll look more at CSF. But just know that glial cells produce cerebrospinal fluid, and that'll make more sense whenever we look at what cerebrospinal fluid actually is. And then know that neurons, are one of the main cells of the nervous system. I'm not going to have y'all know the different types of glial cells, except I think it's crucial that you know what a Schwann cell is, because Schwann cells have a major role in the body. You might have heard about them in lectures so far. And what they do is they surround the neuron, which these guys are kind of like neurons right here, and they will increase the speed of a nerve impulse. So Schwann cells will actually come together and form this thing called the myelin sheath. And we have a slide over this coming up, but this is kind of just a forewarning. Schwann cells will come together. You can see this individual thing right here is one Schwann cell, and they come together and form this whole thing called the myelin sheath, which will increase the speed of nerve impulses and will also help in the regeneration of damaged nerve fibers. So you can see those Schwann cells, and then you can also see that myelin sheath, that entire thing. So if you know anyone or you personally have multiple sclerosis, it's a pretty serious disease. I have a really good friend that I met when I first came to Texas State that has multiple sclerosis. And what it is a disease in the immune system that eats away at the protective covering of the nerves, AKA the Schwann cells in the myelin sheath. So here you can see that individual Schwann cell that comes together to form the myelin sheath. And people that have multiple sclerosis, their myelin sheath, those Schwann cells, are damaged very easily and do not regenerate very quickly. And what it'll do is cause a much slower transmission of impulses. So there are days when my friend cannot walk, her legs will not move. There are days when she cannot lift her arm. There are days when she can't talk. It's because those Schwann cells are deteriorating and they're not quickly regenerating. They're just kind of being damaged. So it takes a long time for one impulse to get to wherever it needs to go. So if you've ever heard of multiple sclerosis, that's what's happening. So now we're going to get into the structure of a neuron. So this will be another diagram I draw on the board that I think would be helpful to you if you drew along with me. I draw a much simplified version of the neuron. It's not, super anatomically, correct. It's not very detailed, but it's how I learned the neuron, and it has helped me to this day. So I recommend drawing along with me to understand the structure of the neuron. So I draw it kind of funky, but it works. So that's how I draw my neuron. So again, neurons are one of the main communicating cells of our nervous system. They are the ones who, have that sense. They sense something, they integrate it, and they send a signal. Let's look at how they do that. Neurons are composed of three major parts: the dendrites, the cell body, and the axon. What are those? Well, let's see. So first, we have the dendrites. The dendrites are branches off of the body that will receive signals. So this is our dendrite, and it receives signals. Receives signals. So the dendrites are the branches off of the neuron that receive a signal. Then we have the cell body. Here is the cell body. You know, inside here we have a nucleus and all those other organelles. We're not going to specifically look at any of those, but this is the cell body. And here we integrate the signal. So dendrite receives the signal, cell body integrates the signal, decides what to do with it, decides if it's actually strong enough to send an impulse to the brain. If it's strong enough, it's going to send the signal down this structure, this whole structure right here, called the axon. The axon sends a signal. So, one more time, dendrite receives a signal, cell body will integrate it, decide if it's strong enough. If it is strong enough, it will pass this point right here at the bottom of the cell body called the axon helix. So that cell body will decide if the signal, if the impulse is strong enough to pass that axon helix. For example, if a mosquito landed on my arm, and this is kind of gross, but it's kind of cool. When mosquitoes land on their arm, on your arm or anywhere on your body, they actually vomit on you. So when a mosquito lands on you, it immediately vomits on you. And in that vomit are chemicals that will numb whatever area the vomit landed on. So the mosquito lands on you, vomits on you, numbs that area. That's why you normally don't feel when a mosquito lands on you, and you don't feel whenever it pricks you with its little nose thing. So because of that, that signal would not be strong enough to pass the axon helix and go all the way down and be sent. It would just, the cell body would decide, oh, it's not strong enough, just get rid of it. Now, if a bee lands on you, bees are a lot bigger and they don't numb you and they actually sting you, your cell body would pick that up and it would pass the axon helix, be sent down the body of the axon, and then it would leave the signal and we're talking about electricity here, so all of this is electrical and chemical. The signal would leave via the axon terminal. So the signal leaves via the axon terminal. It might be electricity, like an electrical impulse, or it could be a neurotransmitter. So one more time, this is our neuron. We have three major parts: the dendrite, the cell body, and the axon. The dendrites are branches that will receive the signal. That signal will be sent to the cell body where it'll be integrated. We'll decide if it's strong enough. If it is strong enough, it will pass the axon helix and move down the axon, be sent out via the axon terminal to either the next neuron, or some neurons are, the length of your entire arm. So the dendrites are in the tip of your finger, the cell body is in the tip of your finger, and then the axon goes all the way to your spinal cord. So these cells can be extremely long. Some are the entire length of your leg. So those dendrites in your tip of your finger pick up that you touched a piece of ice. Pretend this is a piece of ice. The dendrites receive the signal that, oh, I'm touching something cold. It sends it to the cell body. The cell body goes, oh yeah, that's cold. That's, strong enough to send to the brain. It passes the axon helix, travels down the length of the axon to my spinal cord. The spinal cord sends it to my brain. My brain decides, okay, we're touching something cold. And then I have a motor output of realizing I'm touching something cold, and then whether I'm going to take my finger off or endure the coldness. So Nervous system, it's so like not that interesting to me. So sorry if this is not making much sense, you know. The brain. Alrighty, so back to our slideshow. Get the screen share back going. So that was our neuron, dendrite cell body axon. So just a second ago, we talked about those Schwann cells forming the myelin sheath. Now we've talked about the dendrites. You can see the dendrites up there, those branches, cell body, and the axon, with the axon helix up here and the axon terminal down here. So I told y'all that Schwann cells that form the myelin sheath will increase the transmission of an impulse, so increase like the speed that it's sent. So you can see a neuron that does not have those Schwann cells in that myelin sheath, that impulse travels really slow compared to a neuron that is myelinated. So myelination will increase the speed of an impulse. Very important. So here we have 3 structural classes of neurons. I'm not going to have y'all study these, so do not get bogged down. I'll go over them really quickly, but you do not need to study these. So first we have the multipolar neurons. These make up 99% of our neurons. These are the ones found throughout our entire body. Then we have the unipolar sensory neurons, which are going to be like our sensory receptors, and we'll actually see a bundle of these when we look at the spinal cord. We have a bundle of sensory receptors that we'll look at. And then lastly, we have the bipolar neurons. These are extremely rare. They're mostly found in the retina of the eye and in the epithelial tissue of the nose, but very rare. And again, y'all do not need to know these. They're just in case you hear them in lecture. So we can also base our neurons off of that functional class. So I do want y'all to know this, but we've already kind of looked at what this means, so it should make sense. Sensory means afferent impulse, and afferent impulse is something that's arriving at the brain and spinal cord. So what you see is that a sensory neuron will transmit an afferent impulse to the central nervous system, to the brain and spinal cord. It'll arrive. Motor neurons transmit efferent impulses that will be sent from the central nervous system, exiting the brain and spinal cord out to the rest of the body. So sensory afferent, arrive, motor efferent, exit. And then we also have interneurons, which are just that integration site in between. So these would be the most abundant because we have one in between every sensory and every motor. So these are just where we decide, you know, what to do with that information. So now we're getting into action potentials. This is the last part of this slide, and we will have one more diagram to draw here that is very important. So if you have talked about action potentials in lecture, you know that they are just a transmission of impulses. So our nervous system communicates using electricity and chemical messaging. So, you know, we use electrical impulses, and we also use neurotransmitters like acetylcholine, you know, different hormones, things like that. And those will transmit via action potentials. So action potentials are just, that's an action potential. That impulse of energy right there, that transmission of an impulse, that's an action potential. All neurons can only transmit impulses of one strength, but what can change is the frequency and speed of impulses. So let's kind of figure out why. So this diagram, this chart right here is so important. You'll see it on your lecture exam and you will see it on mine. So we're gonna go ahead and draw it out in a diagram form so we can kind of drill it into our brains, make it make some more sense. So here we go. Right, so action potential chart, let's go.
Okay, so our chart. So here is what an action potential looks like whenever we draw that. That's what it should look like when you draw it out. So our chart elements, so our labels. Here, this is a cell or a neuron at rest. There are no impulses, you're just laying in bed, nothing's going on. The cell, when at rest, is at negative 70 millivolts. Negative 70 millivolts is a cell at rest. Negative 70 millivolts is a cell at rest. Whenever something happens, so we're just sitting here right now, B lands on my arm. There will be an impulse. And since it's a B, that impulse will be strong enough to be sent down the axon, and it'll hit this point called threshold, which is negative 55 millivolts. Negative 55 millivolts is threshold. Let's say a mosquito landed on my arm. So we're sitting here at rest, mosquito lands on my arm, not strong enough to hit threshold, returns to rest. So if it's strong enough, it'll hit threshold. If it's not, it won't. So if it doesn't hit threshold, it'll just return right back to rest. If it does hit threshold, real quickly, the cell will be depolarized. The cell will become depolarized. It will depolarize all the way up to positive 40 millivolts. On your slide and in your manual, it says positive 30. That's incorrect. It should say positive 40. Cells, an action potential can only go up to one uniform strength. It can't be less, it can't be more. It's always going to be positive 40. Threshold will always be negative 55, and rest will always be negative 70. It only is at one strength. So, what is depolarization? What does that mean? The cell's being depolarized. What depolarization means? I should have color-coded that. Let me color-code it so it makes a little more sense. So, we hit threshold, the cell is depolarized. What we mean by depolarized is we have a rapid influx of sodium. An influx or an inflow of sodium. So sodium will flow into the cell. I don't really know what the molecule does to the cell. I can't remember. You'll go over that in lecture. But generally, the main thing that's happening there during depolarization is Na plus sodium. We have an Na plus influx, a sodium influx. Sodium flows into the cell, which messes with the voltage, which is why, we have millivolts. Sodium will make it more positive, I guess. And once we hit that positive 40 millivolts, then the cell will go through repolarization. It will become repolarized. So depolarized, sodium flows into the cell, brings it to positive 40 millivolts. Once we hit that positive 40 millivolts, all of a sudden, potassium, K plus,
will start to leave the cell. So we'll have a potassium outflux. A potassium outflux, a K plus outflux. And this will bring the cell's voltage more negative. So it's negative again. Repolarization, sodium outflux. Sometimes the sodium doesn't stop flowing out, and we go into this phase called hyperpolarization. Hyperpolarization. And this is when the potassium keeps kind of flowing out of the cell, but it'll go back to rest eventually. So, a cell is at rest at negative 70 millivolts. A mosquito lands on your arm and it's not strong enough to reach threshold, goes back to rest. Cell is at rest at negative 70 millivolts. A bee lands on your arm, or a roach, or a scary bug. Signal is strong enough to pass threshold at negative 55 millivolts. Threshold at negative 55 millivolts. The cell will become depolarized. It will become more positive, depolarized. It will get up to positive 40 millivolts because we have an influx of sodium. So sodium flows into the cell, makes it more positive voltage-wise. We go through depolarization. Once we hit that positive 40 millivolts, the cell will be repolarized. It'll become more negative because potassium is flowing out of the cell, so we have a potassium outflux. And it'll repolarize back to rest, but sometimes the potassium will continue to outflow, will go into hyperpolarization, but eventually return to rest at negative 70 millivolts. So, know this chart, know how to label it. Everything I wrote on here is important. The number is important, so I could ask you, a cell at rest is at blank millivolts. You would need to know negative 70, not even just 70, negative 70. Or I could ask you, at negative 70 millivolts, a cell is blank, and you would need to say at rest. Or I could put a picture of the cell, or of this chart, and I could say, what's this point right here? And you would need to say negative 70. Or I could say, what's occurring at this point right here? Rest. Or I could say what's occurring right here. What's the upward, upward slant of the chart? Depolarization. What's the downward slant of the chart? Repolarization. What's happening right here? Potassium's flowing out. What's happening right here? Sodium's flowing in. Make sure you know that. You'll see that really heavily in lecture, super heavily in lectures. So it's your best to double study there. Okay. So that was it for our anatomy and physiology of the neuron. So I'm going to go ahead and move on to our next slide. And while I'm getting there, feel free to ask me any questions. And if you need to go get some water, get a little snack, whatever you need to do, we have 1, 2, 3 more slideshows to get through. 3 more. Also, I got this really cool new water bottle. I suck at drinking water because I'm addicted to Diet Coke. So it tracks, you charge it, and there's an app on your phone, and it tracks how many ounces of water you've drank a day. So I'm becoming healthier, even though I never even hit 20% ever.
Where'd you get it from?
From Ross. It was $9 from Ross, so I was like, that's really cool. Why not?
That's really cool.
I love Ross shopping. Yeah, it's so cool. I love this. And then I got a really cool scale on Amazon that was I think $11. And it also connects to an app on your phone. So whenever y'all get into your higher level exercise sports science classes, you'll start to learn how to calculate your water weight percentage in your body, how to calculate your muscle mass, your body fat, all that. So things like this, having data of it is so cool. So, yeah, go to Ross.
Okay. Spinal cord and spinal nerves. So this is chapter 14, the spinal cord and spinal nerves. So let's begin. So the spinal cord, what is it? It is composed of nervous tissue. If you remember, our four major tissue types, we had epithelial tissue, connective tissue, muscular tissue, and now we are to our nervous tissue. So all that nervous tissue we talked about at the beginning is what composes the nervous, or what composes the spinal cord, brain, and all those nerves. The spinal cord functions for reflex actions and transmission of nerve impulses to and from the brain. So it sends impulses to the brain, and then the brain integrates it, and then it sends impulses out to the peripheral nerves. And yeah, we also see reflex actions at the spinal cord, and we'll look at what those are in just a second. So here is a diagram of our spinal cord. You can see the brain is up here, and then this would be the base of the skull. So like our occipital condyles would be right here. And then this is the spinal cord. It starts here and it ends here. So whenever we look at the brain and the next spinal cord, we'll see that the end of the brain is called the medulla oblongata. If you've ever seen the movie Waterboy with Adam Sandler, you've probably heard that word before. The medulla oblongata is the lower end of the brain, and that's where the cord originates. So here is where the cord originates, at the medulla oblongata. I want you to know that. And when we look at the structures of the brain in a second, it will make more sense. But the cord originates at the medulla oblongata. From C3, cervical vertebra 3, all the way down to cervical vertebra 4, C3 to C4, all the way down to thoracic vertebra 1, we have the cervical enlargement. And this contains neuronal cell bodies that serve the upper limbs. So the dendrites are out here. They connect to the cell body in the cervical enlargement, and then the axon will come back out. You don't need to know the cervical and lumbar enlargement, but just so you know. T9 through T12, so, you know, the thoracic middle region of the back contains the neuronal cell bodies, down here at the lumbar enlargement contains the neuronal cell bodies that serve the lower limbs, so your legs and your feet. And then one of the structures I do want you to know is that the cord terminates at the conus medullaris. So it originates at the medulla oblongata, terminates at the conus medullaris. That's a really funky word, so make sure you rehearse that, write it down a few times. It terminates at the conus medullaris. So that's where the cord ends. And then we have extensions off of the cord. So the first extension is the quadra equina, which in Latin means the horse's tail. So the quadra equina, that means horse's tail in Latin. And then lastly, the filum terminale. And what this is just nerves that will connect the spinal cord to the coccyx. So if you've ever been like skateboarding or like rollerblading or anything and you fall on your tailbone, the coccyx, it really freaking hurts. And that's because the filum terminale connects, you know, your sensory to your integratory, and you feel that. So cord originates at medulla oblongata, terminates at the conus medullaris. The quadra equina extends off of it, also called the horse's tail in Latin, and then the filum terminale also extends off of it. So the spinal cord is so delicate. I mean, a tiny little pinch or, you know, injection, anything, just the smallest little anything to the spinal cord can result in paralyzation, death, anything. So the spinal cord is so important, it's so delicate, so we have to protect it. And so we protect it by the vertebral column, so of course the bones of the vertebra. If you remember when we looked at those, we have all those processes coming off of it and that big body. So the vertebral column serves as a major protection of the spinal cord. We also have meninges. We'll look at what those are in a little bit. Cerebrospinal fluid, again, we'll look at that, and fat or adipose tissue. So let's look at how those protect the spinal cord. Oh no, I have the wrong PowerPoint pulled up. Get it fixed so y'all can be able to fill in blanks. Let's see. Here is my spinal cord and spinal nerve. Nope, not that one. Incorrect. Try this one. Oh, I need to organize my laptop so bad. It's so slow. All right, here we go. Okay, so the meninges. What are the meninges? Well, these are membranes that surround the brain and the spinal cord. So we have three of them. We have the dura mater, the arachnoid matter, and the pia mater. The dura mater is the outer tough covering, the outer tough membrane. When we look at the brain in the next PowerPoint, we see that it actually adheres to the inside of the skull. You can see in this diagram, there's the skull, the dura mater, the brain at the bottom. Dura mater in Latin means tough mother. So you can think the tough outer layer. Then we have the arachnoid layer, which is the web-like inner layer, or the web-like inner membrane. Arachnoid means spider, so web-like arachnoid makes sense. And then we have the pia mater, which is the soft, delicate inner layer. Pia mater in Latin means soft mother, so soft mother, so the soft innermost layer. And we'll be looking at these meninges again when we get to the brain PowerPoint, but they also surround the spinal cord. Then we have the fat-filled space surrounding the spinal cord called the epidural space. And its function is for shock absorption. So if you ever jump from something really tall, like if you jump off like a roof of a first-story building, or like down a couple steps of stairs or whatever, that's a lot of like shock to the body. That's a lot of gravity, tension coming down, force from the ground coming up. So the epidural space, that fat-filled space, kind of absorbs some of that shock and takes some of that pressure off of the spinal cord. Most commonly, whenever people hear the epidural space, they think of an epidural injection. These are used during childbirth, during labor. And what it is they inject a chemical into the epidural space right there that will numb you from the hips down. So you will not feel your hips down. And it will stop pain signals traveling from the spine to the brain. When I made this slide, I was totally freaked out because if you look, here is a vertebra, and look how tiny that little space is that they have to stick a giant needle into through your vertebra. Like, that's so gross. So if you remember, we have those intervertebral discs, so they have you lean forward, and I guess they have some method on deciding where to inject, but they have to inject precisely in between those intervertebral discs into that small little epidural space. Oh, that is so scary. So yeah, epidural space is a fat-filled space. Then we have cerebrospinal fluid, which we'll look more at this when we get to the brain, but what it is a fluid that flows through the brain and central canal of the spinal cord to help with protection and then mainly transport of oxygen and nutrients. So it's kind of what helps the spinal cord and brain stay alive. It transports the oxygen and nutrients throughout. And lastly, we are going to look at the internal anatomy of the spinal cord. So I'm not going to really focus on these. I won't ask you any questions about gray matter and white matter, but just to kind of give you an idea of what it is. Gray matter is composed of the cell bodies and dendrites. White matter is composed of the axons, tracks of axons, and more specifically myelinated axons. So axons with that myelin sheath formed by those Schwann cells. Gray matter processes and integrates, it receives and integrates just like the cell bodies and dendrites receive and integrate. White matter sends impulses to and from the brain. So those, remember, axons send the signal. And then lastly, we have the central canal, which is this tiny, tiny, tiny little canal in the middle of the spinal cord, and it contains that cerebrospinal fluid that will transport oxygen and nutrients to the spinal cord and brain. Just breaking down this picture a little more, you can see those three meninges: the dura mater, the outer tough layer, the arachnoid matter, the inner web-like layer, and the pia mater, the innermost soft, delicate layer. You can see that epidural space. You can also see the transverse processes of the spinal cord and spinous process right there. You can see the body of the vertebra, and there's that intervertebral foramina. You can see the white matter, and then you can see the gray matter. I've got a better picture of that. We're not going to look at spinal nerves. We don't have enough time. There are 31 pairs of them, but we're not going to look at them. We are going to look at the classification, however. So whenever you do look at a nerve, so this is our spinal nerve right here. Here's our spinal cord. We have a dorsal root and we have a ventral root. So coming in, the impulse will come in through the dorsal root. It'll pass through this bundle right here called the dorsal root ganglion. It'll be integrated at the gray matter, here's where our cell bodies and our dendrites are, and then it'll be sent back out through the white matter of the ventral root. This dorsal root ganglion right here, ganglion in Latin means a bundle of, so that's a bundle, the dorsal root bundle. It contains sensory neurons, which will, sense that information. It has a lot of sensory receptors there. So I do want y'all to know this, and I have a saying that will help y'all remember it, and it'll help you remember afferent and efferent from the rest of the classifications we looked at. So if you want to draw this or write this down somewhere, it's pretty helpful. So let's look at these. We'll do this memory trick real quick. So the saying is, same Dave. Same Dave. Haha. And what it means is same Dave is sensory impulses are afferent impulses. Motor impulses are efferent signals. Sensory afferent, motor efferent. So this is just looking at, direction and signal direction. The Dave part is looking at the spinal cord section. So dorsal root is going to have an afferent signal, an arriving signal. The signal arrives via the dorsal root. And then ventral root is the efferent signal. The signal exits via the ventral root. And if you remember, dorsal is back, like a dorsal fin of a shark. Ventral root is front, like you vent a lot. Ventral, vent, dorsal, back. So same Dave. Sensory afferent, motor efferent, dorsal afferent, ventral efferent. Hopefully that's like a little trick. Good job. So that sensory afferent impulse will come in It'll pass through the dorsal root ganglion, that bundle right there, and I do want y'all to know that and be able to label it. It'll move into the spinal cord, be integrated, and then an efferent motor impulse will be sent out through the ventral root to the body. And there's kind of just an overview of the spinal cord and all the nerves branching off of it, but you don't need to know. Oh my gosh, I'm getting so tired. So that was the spinal cord and spinal nerves. 2 more to go, and these last 2 are semi-interesting. So I don't even know if any of y'all are still there listening to me. Brain and cranial nerves. This is chapter 13. So this is going to be primarily labeling. So what I want you to focus on here is the structure, where it is. I have a lot of information about the structures. I'll tell you what information I actually want you to know, but with the brain, I'm going to be focusing mostly on being able to label it. So let's begin. So the brain is divided into 6 major regions. You do not need to know these 6 major regions. We have the cerebrum, the diencephalon, the mesencephalon, the pons, the medulla oblongata, and the cerebellum. So those are the major regions, but you do not need to know those. The first major structure of the brain is the cerebrum. The cerebrum is this giant upper portion of the brain. It makes up the bulk of the brain. It is the largest region, and it is divided into the left and right hemispheres. You've probably heard about the left and right hemispheres of the brain. And they're actually contralateral regions. Whatever the right side of your brain interprets, it will deal with the left side of your body. Whatever the left hemisphere of the brain interprets will deal with the right side of the body. If you've taken like psychology or anything, you've learned the details of that, and you don't need to know that for an exam, but that's where the contralateral aspect of our brain comes in. The cerebrum is surrounded by the cerebral cortex, which is gray matter. You do not need to know that. And what makes up the cerebrum is the gyri, or the gyrus, and the sulci. So let's look at what those are. The gyri, or the gyrus, are the mountains, the upper mountain-looking things. And the sulci are the grooves. So those like grooves that go into the brain right there. So I always think like sulci, valleys, the Silicon Valley, sulci, silicon, it kind of sounds the same. So those are like the valleys, and the gyri are the mountains. And I feel like there's a mountain range called like the gyri or the gyrus mountains. So that's how I always remember those. Sulci, valleys, gyri, mountains. So then we have the diencephalon, which is this entire region right here. So let's look at the structures of the diencephalon. So we have the thalamus, which is a relay station for incoming information. Don't need to know that. We have the hypothalamus, which controls the autonomic control center. So all of our involuntary controls of our body are controlled by the hypothalamus, such as, body temperature, tells you if you're hungry, tells you if you're thirsty, tells you if you've drank too much, makes you pee a lot, will make you sweat. The hypothalamus is the main connection between our endocrine system, which is our hormonal system, and our nervous system. So I'm going to save the hypothalamus for endocrine system, which we'll be looking at after the practical. Which, by the way, next week with your practical, just in case I forget to announce this, whenever you're done, free to go. We're not like going to have a lab after too much in one day. So just a by the way. So the hypothalamus will control all of that involuntary aspect of our body. Then we have the epithalamus. The epithalamus contains the pineal gland. So that is something I want you to know. The epithalamus contains the pineal gland. And I don't know if any of you have heard about the pineal gland before. It's a very interesting gland. It's mainly known for secreting melatonin. If you've heard of melatonin before, It is a hormone that regulates our sleep cycle. You can buy over-the-counter supplements of melatonin. They have like 100 milligram, 250 milligram supplements of melatonin that you can take before you go to bed, and they'll help you sleep. Warning, melatonin is known to give nightmares sometimes. Just, if you start having nightmares, if you've been taking it, it might be because of that. But also, the pineal gland was one of the last structures and organs of the body to be discovered. And it has a bunch of very, very, very interesting characteristics. In some cultures, it's called the third eye, or the Descartes seat of the soul. It has a lot of properties when it comes to like psychedelics, lucid dreaming, just the more visual and psychedelic aspects of the mind. I have a lot of interest in the pineal gland, and I'll talk a lot more about it whenever we get to the endocrine system. But the epithalamus does contain the pineal gland, which secretes melatonin, and melatonin is a hormone that regulates our sleep cycle. I want you to know that. Thalamus, no. Hypothalamus, no. Epithalamus, yes. So now we have the cerebellum. So a lot of people have a lot of trouble remembering that the cerebrum is this big, large structure, and the cerebellum is this little small section. Sometimes the cerebellum is called the little brain. So a really, really, really dumb way to remember this, and it is so dumb, it's one of my worst memory tricks, but when I was taking this lab, I could not get down the difference between the two, and this is the only thing I could come up with. So I think of cerebellum. Let me explain it real quick before I give the trick. This structure right here, the cerebellum, contains this tree-looking structure called the arbor vitae, which in Latin means the tree of life. So the cerebellum contains the arbor vitae, which is the tree of life. What the arbor vitae actually is tracts of white matter. So a lot of myelinated axons are in here that are sending signals down the spinal cord into those spinal nerves. And so how I remember that this is the cerebellum and this is the cerebrum, I think cerebellum, Princess Belle from Beauty and the Beast. There was like a part at the beginning where she was running through the forest. So she was running through the trees. So cerebellum, Princess Belle was running through the trees. So the cerebellum is the one that looks like a tree. And then the cerebrum, I just think like whenever you drink a lot of rum, you kind of get like a big head, like a big mouth, and like you just like don't care. So cerebellum, Princess Belle, tree, cerebrum, get a big head. It's that big structure of the brain. That's how I remember those. Super stupid, but it might help you in the long run. What the cerebellum actually does is helps with coordination of the somatic motor function, so our voluntary control, so like our skeletal muscle contractions, and it also stores learned muscle patterns, like playing the piano, shooting a free throw. If you practice, driving a car, if you do those things for long enough, you kind of get muscle memory, and it's stored in the cerebellum. For this, I just want you to be able to identify that this is the cerebellum, and the tree structure inside of it is the arbor vitae, which means the tree of life. You don't need to know the function. So now we're going to look at the brainstem. The brainstem has three portions: the midbrain, the pons, and the medulla oblongata. Sometimes the midbrain is called the mesencephalon, but I like to call it midbrain because that's a lot of an easier word. The midbrain is the superior or top portion of the brainstem, and it contains the visual reflex center, so it'll coordinate head and eye movements. The inferior portion, the pons, or the inferior portion of the midbrain contains the auditory relay center, so like hearing. And then the deeper portion contains a high content of melanin pigment, which is a precursor of the neurotransmitter dopamine. So the deeper portion of the midbrain has functions with dopamine release, which relates to like depression and anxiety and things like that. But for this, just know that, the brainstem is the midbrain, the pons, and the medulla. The pons, what it does is relay impulses between the higher and lower brain regions. It's composed of like conduction tracts, so it kind of just helps the brain talk to itself, but you don't need to know that. And then lastly, the medulla oblongata. This is the lowest portion of the brain stem. And what we know when we looked at the spinal cord is the spinal cord originates at the medulla oblongata. So after the medulla oblongata, we have the spinal cord right here. The medulla oblongata part is the most inferior part of the brainstem, and it has some control over the cardiovascular and respiratory systems, so the heart and the breath, but you don't need to know that. Just know it's the end of the brainstem. And if we were in person, I always play that clip from Waterboy because I love any type of Adam Sandler movie. And I don't know if any of y'all have seen that, but there's like a big funny part where he talks to the teacher and they get in a like argument over Waterboy, and it's just funny. So I love that part. It just makes me so happy. I love Waterboy. I need to watch that again soon. So the meninges, we looked at these with the spinal cord. Remember, we have the dura mater, the arachnoid mater, and the pia mater. The dura mater is, again, that thick outer layer that adheres to the inside of the skull. Thick outer layer. Dura mater in Latin means tough mother. You can see the dura mater right here. Normally in lab today, we would be doing a brain dissection. I'd have each of you walk up to me with a tray, and I'd put my hand in a bucket and dig out a brain and hand you a scapula and some scissors and probes, and we'd be cutting it all apart. And sometimes the brains would have these membranes attached to them, and we could always see the dura mater. Then we have the arachnoid mater, which again is that inner web-like layer. So you can see there it kind of looks webby. Like if we had the brain, I would have y'all rub the top of it and like this white kind of layer would come up. That would be the arachnoid mater. And then the pia mater is that thin, delicate innermost layer, and you cannot see it because it's so thin and delicate. And in the video we're going to watch of the brain dissection in the coming slides, she does say you can see it, but you can't, so just FYI. We also have the ventricles of the brain. These are another structure of the brain. They are 4 chambers deep inside the brain. You can kind of see them right in there. And they contain cerebrospinal fluid, that CSF. And again, that CSF will cushion and protect the brain. And so just a little story real quick. When it was my first semester as an IA, I shadowed under this other IA. She was freaking amazing. She was like 20 years old, graduated high school by like 14 somehow, went while she was like 14 to 17, moved to Spain and taught medical English to Spanish doctors, came back, got her associates as a nurse, or however you get your nursing license, started working as a nurse in the trauma unit, got accepted, decided to get another degree while she was getting that degree, got accepted into like Harvard's medical program, deferred her admissions, and was just taking some more classes at Texas State. She was freaking awesome. But how she became so freaking awesome was kind of a tragedy. So when she was younger, she was in gymnastics, and they were doing stunts on the bar, whatever you call them, whenever you do like a handstand or a flip or whatever on the beam. And she misstepped and she ended up hitting her head really hard on the beam. She remained conscious, you know, she was just laying there. And they were all checking on her, and like she seemed fine, but she had this clear liquid coming out of her nose, and they all thought like maybe she was kind of just snotting because she was crying or something or whatever. And so they didn't pay much attention to it, and all of a sudden, coma for 6 months. She was in a coma for 6 months, and what had happened was one of those ventricles in her brain was ruptured whenever she hit her head really hard. And that CSF was that clear liquid dripping out of her nose. Cerebrospinal fluid cushions and protects the brain, so if that's leaking out, our brain is kind of deforming, shrinking, adding more pressure. And she went into a coma for 6 months. I had a student last semester, when I told this story, the same thing had happened to him. He was skateboarding, he fell off of his skateboard, hit his head, was conscious, felt fine, had some stuff coming, some clear liquid coming out of his nose, didn't think much about it. He was in a coma for 3 months all of a sudden. So make sure if you ever hit your head really hard and you have liquid coming out of your nose, that's cerebrospinal fluid, and you need to go get that ventricle fixed up real quick, because you will go into a coma or go die. So ventricles, don't want to rupture them because they contain the cerebrospinal fluid, which cushions and protects the brain and spinal cord. So this is the video we're going to be watching real quick. This is what we would be doing in lab today. So here is our sheep brain dissection, and just FYI, the sheep brains are a whole lot smaller than the human brains. I have a picture of a human brain right after this, but here is our sheep brain dissection.
Okay, this is our sheep brain. This is the anterior portion of the brain. This is the posterior with the cerebellum and the brainstem. And then this is the superior surface, and this is the inferior surface. We've got our lobes of the brain. We've got frontal. This corresponds directly with the skull. So frontal, temporal, parietal, occipital, and then the cerebellum here, and then the brainstem back here. On the inferior surface of the brain, we can see the cranial nerves. So the first one is the olfactory. So this is the olfactory bulb, and then the nerve runs back that way. This is the optic chiasma. This is where the optic nerves crisscross, and the brain will integrate the images. And then these are all of the other nerves that you can't see that well because of the dura mater. And then this is the pituitary gland. And then we've got the pons, and then the medulla oblongata, and then the true brainstem back here, the spinal cord coming out. These are the meninges of the brain here. So this top layer, this is the dura mater and the arachnoid mater together. And then the pia mater, you can see how this web-like structure is peeled up here. So you can peel that pia mater out of the grooves. So the hills of the brain are the gyri, singular is gyrus, and then the groove is the sulci, or sulcus is singular. I've made a mid-sagittal cut down the center of the brain here, so we'll open the sky up. You can see the cerebellum much better. This branching structure is called the arbor vitae. It means tree of life in Latin. And then here, once again, pons, medulla oblongata, yes, and spinal cord. So this kind of circular region of the brain, this is the thalamus. Epithalamus is up here, this little guy right here, that's our pineal gland. And then the hypothalamus is kind of this area down here. You'll see this is where the optic chiasma and the pituitary gland is. And then this little bridge, this is the corpus callosum. And this area is the cerebrum. And what am I missing? In the arbor vitae, the tree structure, the lighter structure, that's white matter, and the darker structure is the gray matter. And that is the brain.
So yeah, that's just a little preview of what we would have been doing in lab today. All those structures she said, I definitely won't have you know all of them, because some of them you need to actually use your finger to kind of feel and see how that's the hypothalamus, that's epithalamus. I think that's kind of confusing. This is a human brain, so it's a whole lot bigger. Those structures, those gyri and the sulci are a whole lot easier to see. So here's what I would want you to know for your practical, but not all of these. these. So if you have like a highlighter right now, I'll tell you which ones I would like you to know. So definitely know that big upper portion of the brain is the cerebrum. Know that that little tree structure is the cerebellum, and then know the arbor vitae, that tree of life structure. I like the corpus callosum because it's easy to identify. I always think of like there's this big bridge in Corpus Christi called the Corpus Christi Bridge. So you can think corpus callosum, that like bridge structure. And then just know that it goes pons, medulla, spinal cord. Midbrain's like right in here, but you know, it's too hard to see. You don't need to know hypothalamus, thalamus. You don't need to know optic chiasma or the pituitary gland or the pineal body. Those are just really hard to see unless you have the brain right in front of you and you can kind of use your finger to mess around with it. So cerebrum, cerebellum, corpus callosum, pons, medulla oblongata. That's about it. Cool. All right. Oops, my chat was pulled up. Can you talk slower, please? Yes, I am so slow. I'm sorry. I'm so tired. I feel like I'm zooming through this. Yes. Thank you. Okay, so this is our last PowerPoint. So let me go ahead and close this one up. So we are to our last PowerPoint. Woo, I'm so exhausted. I'm gonna have no voice tomorrow. So last one before practical number 1, reflexes and special senses. This is chapter 15 and 16. So what is a reflex? A reflex is a rapid, predictable response to a specific stimuli. We have a neural pathway that makes these happen called reflex arcs, and we have 2 of them. We have autonomic reflexes and we have somatic reflexes. Anytime you hear autonomic, it's going to mean involuntary. Anytime you hear somatic, it's going to mean voluntary. So autonomic reflexes are going to be dealing with the autonomic nervous system, helping with those involuntary actions like blood pressure, heart rate, salivation, digestive functions. Somatic is going to be through the somatic nervous system, so our voluntary nervous system, working with skeletal muscle stimulation. And then we also use these reflexes to test the integrity of the nervous system. So let's look at these 2 specifically. So our somatic reflexes, our voluntary reflexes. These are used to check the integrity of the nervous system at the L4 level. You don't need to know like the L4 level. Honestly, I don't even think I have much about reflexes on the exam at all. Maybe I have like know the definition. So we've all probably heard of the patellar tendon test, or maybe gotten it before when you go to the doctor and he hits your knee with that weird probe thing. And what you're hitting is the patellar tendon. We all know that bone in the knee right there is the patella, and there's a tendon that runs in front of it called the patellar tendon. And this can help us figure out if you have like a herniated disc, if you have nerve injury, any type of like compression of the spine. But I don't think I asked you much on that. Another somatic reflex test we have is the Babinski test. This is used to rule out damage to the central nervous system, so to the brain and spinal cord. And what happens is like the doctor would run a pin up your foot. The normal response would be that the toes would go down like flexion. They would kind of curl in. The abnormal response would be that they would go up and flare out. So that could indicate some type of damage to your brain or spinal cord. If y'all remember, I told y'all I was in a really bad car accident when I was in high school. I actually lost my Babinski reflex. Whenever a doctor does that pin up my foot, my toes don't go up or down. They kind of just stay normal. I, of course, feel the pin and it tickles, but I don't have that reflex, which is very minor. It's not like that affects me or anything, but that did show that there was some damage to my spinal cord. So those were our somatic reflexes. Now we're going to look at our autonomic reflexes. So the first one is our pupillary reflex. So if you've ever had like a doctor shine a flashlight in your eye, what they're doing is testing for damage to the brainstem and also for sensory and motor functions of the eye. And what should happen is your pupils should constrict or get smaller when the bright light shines in it. If the pupils don't constrict or they constrict just like minorly or not much at all, that could indicate that there was some type of damage to your brainstem, which is in the lower portion of your neck. So maybe if you've had like a neck injury or if you go in the doctor complaining about neck pain, they might check that pupillary reflex to see, do they have some type of damage? So that's what they'd use that for. So those were the 3 reflex tests we'd look at. And if we were in lab, we would have used the flashlights. We would have had the little knocking tools and all that to do those tests. There's also more reflexes. There's the bicep reflex. You can hit a tendon right in here and it'll cause your fingers to come in. There was definitely more reflexes that we would have looked at if we were in person, but they're not too important. So those are reflexes, and just to give you an example of a real life reflex, it would be like if you put your hand on, let's say you're cooking and you're using the burner. Let's say you put your hand on the burner. What would happen is you would, a nerve would sense it. It would send it to your spinal cord, and then your spinal cord would send out the response to pull your hand away. It wouldn't go from your nerve to your spinal cord and then to your brain, where your brain's like, oh, we're touching something really hot. Now I'm going to send an impulse. It skips the brain and just goes straight to the spinal cord. So you never think like, oh, pull my hand away. Your hand automatically is going to be pulled away if you touch something really hot or you bend your body in a weird way. So a reflex is whenever you don't use your brain to perform an action. Now we are to our last chapter, special senses. This is chapter 16. So we have 5 special senses. We have 5 special senses in the body. I don't think this is written down anywhere, so you might want to write them down. Our 5 senses are smell, smelling, touch, touching, vision, or seeing, taste, tasting, and balance, equilibrium. So sight, smell, touch, balance, and taste. Sight, seeing, smell, smelling, taste, tasting, touch, feeling things, and equilibrium or balance. Those are our 5 special senses. So first we'll look at hearing. So hearing is done via the ear, and the ear also deals with our balance. And I'll explain why in just a second. All these that are highlighted, you don't need to know all of these. Once I get through the explanation of these, I'll tell you which ones I want you to identify. So whenever we look at the outer ear or the external ear, that's this whole section right here. First, we have the pinna, which is the satellite dish structure. So what catches the sound waves, the pinna. The external part you can feel, you can move it around. It's made of cartilage. That's the pinna. If you remember when we looked at the skull, the bones of the skull, we had that hole called the external auditory meatus. And that's so that external auditory canal can intercept those sound waves and send them to our brain. So this is that external auditory canal or the external auditory meatus. Here's the skull and here's that hole. So our pinna intercepts those sound waves, it catches them, sends them down the external auditory meatus, and they hit the tympanic membrane. The tympanic membrane is most commonly known as the eardrum. So know that the tympanic membrane is most commonly called the eardrum. And what happens is those sound waves will hit the tympanic membrane, and the tympanic membrane will vibrate and cause mechanical vibrations that will turn into sound or something. So pinna catches the sound waves, send them down the external auditory meatus, hits the tympanic membrane, which will vibrate and convert those sound waves into vibrations. Then we get into the middle ear. So now we're into the middle ear. And inside the middle ear, we have the ossicles. The ossicles are the 3 smallest bones in the body. I want you to know that. The ossicles are the 3 smallest bones in the body. I'm not going to have you know which is which or the name of the 3. We have the malleus, the incus, and the stapes. I'm not going to have you know them, but I do want you to know that there are 3 ossicles, and they are the smallest bones of the body. We also have the auditory tube that connects the middle ear to the pharynx. Whenever we get into the digestive system and the respiratory system, you'll learn that the pharynx is the throat, and it's actually connected to the ear. So anytime you have drainage, like if you have like a cold or anything and you're having drainage, that's actually earwax draining down from your ear into your throat. So that's real fun. So yeah, our ear is connected to our throat. So we have the eardrum, the tympanic membrane, the ossicles. Next, we get into the inner ear or the internal ear. And we have these 3 circular structures called the semicircular canals. These are the semicircular canals. And these are what help us with our balance and our equilibrium. So inside these 3 bones, there is fluid and bubbles. There is 1 bubble inside of each of these bones. And those are what dictate our directional sense. So I don't know if any of you have ever gotten car sick before or gotten vertigo. That is due to the semicircular canals. So those bubbles, whenever you move, they'll move. Whenever you move this way, they'll move that way. They're gonna move wherever your body is moving. So if you get on a bus, like one of the Texas state buses, and you're sitting facing this way, but the bus is moving that way, those bubbles are gonna sense that you're moving in the forward direction, but you're actually moving in this direction, and those bubbles can start to get lodged. And whenever they get stuck, that's when you start to get that really sick, car sick feeling. And when I get it, I get really nauseous, I get like a stomach ache, I don't feel good in the head, like you just feel nasty. If you've ever gotten vertigo, which vertigo is extreme nausea that is really hard to come out of, it takes a couple minutes or an hour or so, and I've never had vertigo, but my mom has had it. That's when one of those bubbles gets stuck and it won't get unstuck. So you're like trapped in this like disoriented state. So those semicircular canals, I don't fully understand the bubbles. I'm not a hearing major, but those are what is responsible for like equilibrium and balance. And then lastly, we have the cochlea, which is this snail shell looking structure right here, like Gary from SpongeBob. This is the cochlea, and it contains hair cells, cilia, that will convert those mechanical vibrations further into auditory. I don't really understand the ear. I don't have much interest in it, but yeah. So the ones I would have you label on this picture would be the pinna, which is the satellite structure, the pinna, the external auditory meatus, the tympanic membrane, the ossicles, the semicircular canals, and the cochlea. You don't need to know like the vestibulocochlear nerve or the auditory tube. Just know pinna, external auditory meatus, tympanic membrane, ossicles, semicircular canals, and
Wait. Can you say that slower? Sorry.
Sorry. Pinna.
Say it again. The what?
Pinna.
Oh, pinna.
N-A. I know it's a super weird word, which is like the satellite structure you can actually see and feel. The external auditory meatus, the hole, the tympanic membrane, which is the eardrum, the tympanic membrane. The ossicles, the 3 smallest bones of the body, the ossicles. Oops, let me pull my chat up. Sorry, sometimes my chat disappears. Yeah, I get so car sick on the Texas State buses. I just won't ride them anymore. I take my bike everywhere. The ossicles, the 3 smallest bones of the body. The semicircular canals, those help with equilibrium. And then the cochlea, the snail shell looking structure, like Gary from SpongeBob. Those are what I would want you to be able to identify. Function-wise, know that the ossicles are those 3 smallest bones. Tympanic membrane is the eardrum, and the semicircular canals, equilibrium and balance, car sickness, vertigo, things like that. So that was the ear. Lastly, we're gonna look at the eye. So let's get into the eye and then we will finish up. So, I'll tell you which ones I want you to know. The cornea is not one of them. The cornea is the clear anterior part of the eye that covers the iris and the pupil. You don't need to know that. It's just like this clear flap right here. The sclera is the white portion of the eye. So the white part that you see, you know, the middle and the outside part, that's the sclera. The iris is the colored circle of the eye. So like the colored, whether it's brown, green, blue, whatever, that's the iris. A way I remember that is like there's a flower called the iris. So I always think just like flowers are normally colorful, so iris color, the colored part of the eye. Then we have the pupil, which is the black dot in the center of the iris. So that black hole right there is the pupil. And then we have the choroid, which lines the interior of the sclera. It's like this muscle right here. You don't need to know choroid, but no, sclera, iris, and pupil. Sclera is the white part, iris is the colored part, pupil is the black dot in the center. And then all these highlighted things you don't need to know. It was just on the picture I found for the PowerPoint. Then we have the retina, which is back here at the back of the eye. Don't need to know this, but it contains sensory receptors sensitive to light. We have the optic disc, which is right here. It's known as the blind spot of the eye. It's where the optic nerve comes into the eye and meets the retina and causes a blind spot, you know, wherever it is, like right here. We have the lens, which is this marble-like structure. If we were doing the eye dissection in person, what would happen is the eye dissection is one of my favorite parts of teaching the lab. I think it's so much fun because we have these big old cow eyes that we dissect. And so everyone would get a cow eye and scalpels. And whenever we watch the video of the cow or of the eye dissection next, you'll see that they're really hard to puncture. The eye is almost completely filled with fluid. So if you were to take a pencil, which is this super disturbing, but if you were to take a pencil and shove it in your eye right now, a bunch of fluid would come out and it would just be like this little flap of tissue. The eye, a lot of people think it's filled with like muscle or something like that. No, it's filled, this entire inside portion is fluid. So whenever we would do the eye dissection in person, since the eye is so thick, because you know it is filled with fluid, we don't want it to be able to puncture easily, so it's really thick to cut through. A lot of people take their scalpel and they're pushing it in, and then all of a sudden, while they're squeezing the eye to puncture it, fluid literally like would explode all over them, and it would be freaking hilarious because some girl would always get in her hair and like have a panic attack because she got, you know, aqueous humor, which is the fluid of the eye, or the vitreous humor, all in her hair, and it was always really fun. So yeah, normally the eyes, when we do the dissection, they squirt, and so that was fun. But yeah, so the eye is really just this flappy, fluid-filled thing. It's real strong on the outside, but inside it's filled with fluid and this little marble, and the marble is the lens. The lens is what
is responsible for making you have to wear like glasses or contacts because it's what focuses images. And what we'll see when we look at the dissection is it's kind of oniony, like you can peel it a little bit. And that's what they do when you get laser eye surgery. They like kind of scrape it off to make it more focused, things like that. And just something really odd. Whenever I did the eye dissection with my students one year, I had a student ask me if he could take the lens home. And I let him. I'm not sure what he did with it, but it was pretty random. So here's our eye dissection. Let me real quick tell you what I would want you to be able to label here. Pretty much what I would want you to be able to label is just the pupil, the sclera, and the iris. That's about it. So the most basic part of the eye. So yeah, let's watch our eye dissection.
All right, so this is our eyeball. This is the anterior portion, right, with the cornea on the front. The sclera is the white part of the eyeball. I've removed all of the fat and the muscles here, and you can see the optic nerve on the back. So I'm gonna cut this guy open.
So yeah, she knows how to do it right, but normally a student would be squeezing it really hard to get it, and fluid would like explode out of it. It'd be really funny.
And I'm just gonna make a frontal incision to separate the anterior and the posterior portions of the eye. So the first thing you notice is this jelly substance. This is the vitreous humor at the back of the eye. This allows the eye to maintain its pressure. I'm going to take that all out. Okay, and then this is the back of the anterior side of the eye. So this that we see, this is the lens. I'm going to take that out. So the lens is hard. It grows in layers. So if you crack it open, you can see it's very onion-like because it grows in these outward layers. This is why if you get laser eye surgery, they say that it can only be temporary because your lens does keep growing. So, and then the pupil, that's the hole that you can see through, right? This is where light enters the eyeball. On the back of so this area here around the pupil, the colored area of the eye is the iris. On the back side of the iris, you see this is the ciliary body. It's very ridged, right? And then the aqueous humor is in front of the lens between the pupil and the eye, but it's so small that we couldn't really see it. And it's just more of that jelly-like substance. Okay, on the posterior portion of the eye, we have the choroid here. This choroid, this is a cow's eye, so the choroid is covered in a special layer called the tapetum lucidum.
So real quick, this isn't in your slide, but I would like you to know the
This
structure of the tapetum lucidum. If you've ever like taken a picture of like an animal at night, like your dog or your cat or like a deer, and in the picture there's like their eyes look like blue or red sometimes, that's reflecting the tapetum lucidum, and that actually gives animals night vision. So that's why whenever you take a picture of an animal, if you have your flash on, it looks blue. And it's actually like this really pretty like pearly-like structure in the back of animals' eyes. So the tapetum lucidum, whenever the video is over, I'll spell that out.
Better eyesight at night. And it also creates that effect called eye shine. When you shine a flashlight and you see the eyes reflect back. And then this that has partially detached, this is the retina. Where the retina is attached at that point. This is where the optic nerve is coming out the back. It's the same point. We call this area of the eye the optic disc. And then you'll see this little dot over here. This is the fovea centralis. This is the spot on the eye that is responsible for that very focused vision that you have. So cows like us only have one fovea. And then the optic nerve on the back. So that is the eyeball.
I miss dissecting things so much. So yeah, that's the eye.
All right.
So I'm going to spell out tapetum lucidium for y'all real quick because that's a weird word. And again, that functions for animals' night vision. So the tapetum lucidium gives animals night vision. T-A-P-E-T-U-M, tapetum lucidium, L-U-C-I-D-I-U-M, tapetum lucidium. Alrighty, so I think we have 3 more slides to get through and we are done. So testing for visual activity. Most of you have probably used one of these before in your life. This is the Snellen eye chart. And what this tests for is whether you are farsighted or nearsighted. So if you've been told you have 20/20 vision, what that means is that at 20 feet, you see what the average person sees at 20 feet. So 20/20, you see at 20 feet what the average person sees at 20 feet. If you're 20/40, that means at 20 feet, you see what the average person sees at 40 feet. So you have really good vision if you're 20/40. Or sorry, really bad vision. Sorry, that was wrong. 20/40 is bad vision. So at 20 feet, you only see what a regular person sees at 40 feet. So they can see a lot farther than you can. And then if you're like 20/10, that means at 20 feet, you can see what the average person sees at 10 feet. So you have really good vision. I won't ask y'all about any of that. That's confusing. But that's what this test is used for, farsighted and nearsightedness. Then we have tests for astigmatism. So an astigmatism is an irregular curvature of the eye. So if any of y'all have an astigmatism, one or two or more of these lines probably look really blurred to you. So if you like cover one eye, the 2 and the 8 might look like one individual line, like those lines will look really dark or blurred together. And that's because you have a curvature on your cornea that is causing blurred areas. So an astigmatism is an irregular curvature of the eye. Yeah. And then lastly, we have the Isharia test, which tests for color blindness. I've never had a colorblind student in my entire teaching career, and I've taught 6 semesters now. And so my dad is colorblind with red and greens, so he would not be able to see that 8 in the middle. Like he can't see the color red of a red light. He knows it's red because the order, but like he can't see the color red very well, or orange. So he would not be able to see that 8. So people that are colorblind can see certain colors. They're not like seeing black and white, but you know, they can't see certain things. My dad and his brother are the same. Yeah, I know. And I have this really funny picture. It says something like kind of inappropriate in the middle. And I want to use it so bad whenever a student's going to be like, I'm colorblind. And I put it up and everyone like laughs and they like don't know what it says. And so I'm like, oh, I need a colorblind student so bad to play that trick on them. But whatever. So yeah, that's the color blindness test, the Isharia test. We are done. Oh my goodness gracious. So let me make a last few announcements and then we are free to go. That is the longest lab we are going to have this entire semester. There will be no more long labs after this point. So thank you all for sticking through with me. If you are still there, it looks like we've lost a few. So let me head back to Canvas to show y'all what your lab report is for this week. I think it's a pretty fun lab report. I'm excited. And I'm gonna extend the deadline so you can turn it in after the practical if you'd like. I think it'd be helpful to use before the practical, but I'm gonna extend the deadline to, you know, the next week. So it'll be due in 2 weeks from yesterday for y'all. So let me go back to today's module. And again, I think this is just like kind of a fun lab report. I think it's good to prepare you for future ESS classes. So what I'm gonna have you do is attach your outline as usual. And then from the following list, I want you to look up exercises for each of these muscles, 12 of them. Create a Word document of an exercise plan, adding pictures of the exercise and, that should say what type of movement is occurring. So the example I have here is like, look up the triceps brachii, look up an exercise for it online, and then just talk about the movement, flexion and extension is what's going on there. And that's easy to know because in most of these exercises it says tricep extension, which is, of course, flexion and extension. For the gastrocnemius, I have calf raises, so you just attach like a picture. And he's obviously doing dorsiflexion and plantar flexion. So, do 12 of those for these 12 exercises. It sounds like a lot, but it's not bad. And then lastly, I want you to draw a flow chart of the nervous system divisions, which if you drew them out with me today as I did them, you have already completed all this. So you would draw out like CNS, PNS, and then, motor, sensory, and then somatic, autonomic, and parasympathetic, sympathetic. You can do that in Microsoft Word too if you'd prefer to do that. Also draw out a flow chart of the spinal cord structures. So originates at the medulla oblongata. Then we have the cervical enlargement, the lumbar enlargement, the conus medullaris, the quadra equina, and then the phylum terminale. So you just draw, words. You don't have to draw the structure, but just draw a flow chart of how that works. And then the last two, draw the neuron and label the structures. So just like how I drew the neuron on the board today, draw the dendrites, draw the cell body, draw the axon. And then, I think it always helps to just write, that receives, that interprets, that sends. And then lastly, just draw the action potential chart that we drew earlier and just draw the little, things on it. So that's your lab report. It'll be due the week after the practical, so you'll have time. I definitely recommend doing it before the practical because it'll help you in your studying. If you're still there, I'm going to show y'all, and now I'm kind of just making announcements, but I'm going to show y'all how I would study when I took this lab for my practicals. So what I would do is I would take the study guide that they gave us and I would create my own PowerPoints based off of so you can see what I mean. So I would just pretty much copy and paste the slides into my own slideshow, and then I'd put blocks over the, it's not picking up, I opened it. So this was for a different practical we had, but what I would do is I'd just copy and paste and I'd put blocks over what I knew was important. I'd number the blocks and create a key, and then afterwards I'd go through the PowerPoint with a blank piece of paper and I'd say, okay, number one, function of the respiratory system to act as the site of blank and blank exchange. And I'd go, okay, I know that is CO2 and O2. And so whenever I got those right, I would leave it and then I'd cross it out. If I got it wrong, I'd highlight it and I'd know, okay, I need to focus on that. I need to know that, the vocal cords, whatever. And then once I mastered a slide, I would just put a smiley face over it. I found this studying method really effective because it helps you visualize your notes really well. You can think back like, oh, that was that slide and that was that bullet point. I know this. So that's how I would study if you're looking at tips to study. But there's definitely a lot of other ways. But again, this is a practical you have to study for because it is fill in the blank. There's no guessing here. So you're going to be typing in all of your answers. So please study, and I'm totally available to help y'all, and I hope y'all can attend the review on Wednesday. We'll play games, and there will be extra credits. So that's all I have for y'all today. Thanks for hanging around.
Will you send out a reminder for the review?
Yes, I'll definitely send like a group me reminder, and I'll probably send an email with the Zoom code.
Okay, thank you.
Huh? All right, thank y'all. Y'all can log off or ask me questions as you please. I'll be here chilling.